Before mitosis, the cell enters an interphase where it's enclosed in a nuclear envelope. And it has a nucleolus and chromatin and a pair of centrosomes. Before the next stage of mitosis or prophase can start, the centrosomes must be copied and moved to opposite sides of the cell. Then the nuclear envelope and the nucleolus disintegrate. Oh, that's wrong. Then the chromatin condenses into chromosomes made up of tightly coiled histone proteins. The chromosome is made up of two sister chromatids and are joined at the centromere. Spindle fibers then come into the cell and connect to the centromeres. The cell has now entered anaphase, where it has elongated into more of an oval shape, and the chromosomes have broken apart, and the sister chromatids have now become daughter chromatids, and each side is being pulled by the centromeres and to the respective sides of the cell, and these are still together, but they'll be broken apart and pulled to each side and by the centromere, and so they'll be magnetically pulled, and then the cell will split. During metaphase, the kinetochore of the chromosomes line up on the spindle fibers, and the spindle fibers begin to pull from the centromeres. After anaphase, we enter telophase, where a cleavage furrow begins to form, and the cells begin to become their own, and the spindle fibers retract, and each cell have their own DNA. So no more spindle fibers. And each cell has their own centrosome, and each cell begins to make their own nuclear membrane. And their chromosomes become chromatin. And they each form a new nucleolus. And then cytokinesis happens, and they close off, and they form two completely new cells.